And I highly encourage our speakers and others to put their comments and suggestions in the chat space to just make sure that we will be able to continue discussions in the chat space and move forward with the discussions, and then we will have time to kind of discuss about the questions that we already have in the chat space. So the next series of speakers that we have are going to discuss about some of the other aspects of those responses in animal and kind of cellular models. And the next speaker would be Dr. Ivan Alekseichuk. So, Ivan.
Thank you. We can go straight to the next slide. I'm Ivan Alekseichuk, University of Minnesota, and I would like to talk about dose response and transcranial alternating current stimulation, what we know about it today. First, I want to define what I mean by dose and response and what we are seeing here. As we heard today from Dr. Lisenby and Dr. Petychev, dose and brain stimulation can be understood very comprehensively in terms of strengths of pulse or stimulation and frequency and electrode montage and many other parameters, and as a delivered dose, as an electric field. But in the first approximation, we can look at delivered electric field and just the magnitude of electric field to simplify the problem and keep other parameters as a type of stimulation outside of it for a moment. And then about the response. So we heard also today from Dr. Bigson that response to electric stimulation basically starts at 0, and there's always way to go lower in a dose if you assess the induced changes in the membrane potential, which is a primary target of stimulation. However, on a dose response curve, there are a few points of interest, and one of the points that I will talk today is a minimal effective dose, or MAD. That is such a dose that delivers a response that propagates to the next step of the physiological cascade or next level of organization. For example, changes in membrane potential propagate to changes in neural spike timing or other processes. So if you look at available data in all experimental studies, recruit wisdom of the crowd to see how different people define it, slightly different, use different experiments, but what is the summary? We can find, or at least we found, 16 studies from 1984 to 2022 that use alternating current stimulation and estimated electric fields in neuronal population, and looked at some direct neural response in cells, cellular slices, anesthetized rodents, freely behaving rodents, and non-human primates. There is no such data in humans, unfortunately, so far. But anyway, we can look at these studies and we can ask ourselves the question, what would be the minimal effective dose in this study? And we can make an assumption that if a study looked at number of different doses, so number of different induced magnetic fields, there is a magnetic field at which a particular study saw the response. That would be like minimal response, and at least one unit of observation. That would be effective dose in the definition of the study. And then there is a previous dose that they delivered where they haven't saw response. That would be ineffective dose. So our true minimal effective dose lies somewhere between these 2 points. We can assign probability distribution, such as beta probability, to these boundaries, and conduct a meta-analysis and see where the minimal effective dose actually will be. And on the slide, you see a cumulative probability of inducing minimum detectable response due to TACS or some form of alternate current stimulation. And what we see here is that the study pretty much split it into groups. One group is awake and behaving rodents and non-human primates. Other group is brain slices and anesthetized rodents. Awake and behaving mammals in red require much lower dose, so they are much more sensitive. And at already 0.3 volt per meter, we see 80% chance of inducing minimal detectable changes in neural behavior and some spike timing or number of spikes. And the way how we can use that is by integrating with modeling, obviously, in the future. The one thing that keep in mind when integrating modeling is that typically we just run a model and estimate peak of electric field, but that may not be a necessary great approach, because not all electric field equal, assuming that we foremost stimulate pyramidal neurons. We should look more on tangential component of electric field and electric model, and then ensure that the dose that we want to receive, that we want to deliver, is not just at the maximum point there, but across the whole region of interest. So that's something that should be embedded in future modeling or in the future strategies for appliance. And of course, the question of getting such data in humans that have different site architecture and different neuronal and neurotransmitter dynamics is crucial. I want to thank Alex Opitz's lab where I'm working, and NIMH and Brain Behavior Research Foundation for support.
Thank you very much, Ivan. Thank you very much.